All right, so getting started, we're going to talk about kind of our smooth muscle today. So what's different about our smooth muscle, as you can maybe tell by the name, is that it's not striated. So it doesn't have regular sarcomeres like our skeletal muscle did. And so its muscle tone determines the degree of contractibility. And our smooth muscle is found in our blood vessel walls, digestive system, respiratory system, bladder, glands, and follicles. And up here, we can see that this is our skeletal and this is our smooth. And because it has that pre and post, it's not all or nothing like our skeletal. Or like it has those graded contractions. So it's not all or nothing like our skeletal. So moving down here, this is just a good depiction of the shape. So it says it's spindle shaped with one nucleus. And we can kind of see that if we're to highlight one here, you can zoom in and kind of see how it's kind of spindly and it has one lone nucleus in there. So moving down here to the next slide. So we see that it's a little different. So we see that spindle nucleus or that spindle shape, but it also still has those the myosin and actin, those thick and thin filaments, just like the skeletal. However, it's arranged in a cross hatch. So that's why there's no striations. And we see that here with this pattern, this cross hatch pattern that we see. And then down here, we see that our thin filaments are anchored to our dense bodies. And so that's kind of right here is our anchor. And so that's different from our C lines, which is for our skeletal. All right, so moving down here. Our contractions are not like a nice straight line like our striated muscles, our skeletal muscles. So those are like those sliding filaments, a nice straight line. But these kind of ball up as we see. This is called kind of like blebbing. They kind of bleb into one kind of ball, I guess. And so it definitely has like a bigger shape change here. This is just another depiction of that blebbing, and you can kind of see how they get bigger. They're a little less spindle shaped here. And then the last thing I want to mention is that there's no troponin. So only your phosphorylated myosin can bind to your actin. And they do that utilizing these light chains. So these are sites that can be phosphorylated. And once they are phosphorylated, it causes a shift in cross bridge, which allows that blebbing to occur.
And that blebbing occurs because of the shape change that happens. After that shape change, the myosin is allowed to bind to that actin. That binding allows like the ATP to be hydrolyzed, and then that is what makes the muscle shorten and the cross bridge move, which is that what we know as our muscle contraction.